So let's draw. Let's draw a big mitochondria on our paper here. So not all of the steps of cellular respiration, you guys, not all of the steps occur in the mitochondria. Griffin. MRH. The mitochondria, there's the glycolysis that occurs outside of the mitochondria. So when you're thinking about your paper, because first I got really stressed about how big to make the mitochondria and if they're going to have enough room. I'm going to put everything that we draw up on the board in one picture, so it's not like I'm going to do part of it and erase it and so on, so you'll need more room. So I'm going to leave some space over here because I'm going to do glycolysis over here. But I'm going to draw a big old mitochondria, which is bean-shaped. So we have our bean-shaped mitochondria like this. All right. So you can see how you can take a good portion of your paper up, but you want to leave. You don't want this to go all the way to the edge because I'm going to draw some things over here. And then that's the outer membrane of the mitochondria. Remember, the mitochondria has two membranes. So there's an inner membrane that is folded, and that's called the cristae. And this, when we draw the cristae, because of the different phases of it, of cellular respiration, we need some space between the inner and outer membrane. So I'm going to draw it here like this, so it's curvy like this. All right. But you need to have enough space in between here. All right, so that we can draw some things in here. All right, and so so you have to have some space inside and some space out here. All right. So the let's label here. So what would this space outside of here be? What would this be?
Cytoplasm or cytosol?
Cytosol or cytoplasm. I'm going to write this up here. So cytoplasm or cytosol, either one. And then, hopefully, we know that this is the mitochondria, but let's just write it here, mitochondria. For this, we need to know the spaces, or the names of the spaces in the mitochondria. So what is this space right here called?
Mitochondrial membrane.
Well, this is the outer membrane, this is the inner membrane. What is this space in between?
The matrix. Mitochondria.
That's this part here. So the matrix is right here. So I'm going to add this. All right. So the matrix is all this space inside. So this space is here. The name actually sounds exactly like what it is.
Intermembrane space.
Intermembrane space. It's the space between the two membranes. All right. So intermembrane space. I'm gonna label it here, and then because we're gonna draw what happens in that space over here. So this is all the labeling part. So we just know where everything's at. And before we get started, at the top, I'd like to put the equation for cellular respiration. So at the very top of your paper. So I'd like you to see if you can do that. So can you write the reaction of cellular respiration, what goes in and what goes out? Alright, so hopefully you were able to say glucose goes in. So glucose is C6H12O6. And then what's the gas that goes into cellular respiration? Oxygen. So it's O2. Then we have, it yields what two things? Yeah, carbon dioxide and water. So in both of these, kind of like waste products, they're not the main purpose of this reaction. This reaction is what, exer or endergonic? This is exergonic reaction. This is a very exergonic reaction. It yields negative 686 kilocalories per mole of glucose. And so it's a very exergonic reaction. Remember that negative in front of it means that it releases energy. So I'm going to add here so that we think about, when we think about cellular respiration, that this is an exergonic reaction. So I'm going to put here plus energy to do what? What's the, when the energy is released, what's the use or the purpose of that energy release? What is it used to make? ATP. So I'm going to put here energy to make ATP. All right, and that's what we want to get out of this. All right, so that's our end purpose in mind, is to make ATP. So there are three major steps in this process. So this arrow here is not just one reaction. There are many, many reactions to get to these end products. And so that's what I'm going to try and pull together for you today. So that first step in the first series of reactions does not occur inside the mitochondria. It occurs in the cytoplasm, and that first set of reactions is called glycolysis. All right, so therefore I'm going to write some things out here. I'm going to write it in purple, all right? And so some people like to write the same color I do, but it really doesn't matter. All right, so first step, step one, is glycolysis. All right, with these reactions, we don't have to memorize all these intermediates in these reactions and so on. We need to understand big picture. So, and even the big picture may seem like it's a lot of stuff. All right, so glycolysis, what molecule do we start out with? Glucose, right? So, so we start out with glucose at the very beginning. And glucose is C6H12O6. So big picture, I want us to think of it, it's a 6-carbon compound, it's a 6-carbon molecule. Put this up here, because as we go over this with glycolysis, so I'm going to refer, we're going to kind of oscillate back and forth and look at your packet that you did yesterday. So I'm going to keep this up here as we go over this and relate it to that. So we start out with glucose, and really we end up with, does anybody know what we end up with at the end of glycolysis that's going to go to the next step? What do we end up with? 2-pyruvate molecules. In order to get to 2-pyruvate molecules, that's our end product. And these are, does somebody remember how many carbons they are big? Along? 3. All right, so it's like just taking glucose and breaking it down in half, okay? So we have 3 carbons in those. So therefore, the 6 carbons that were in glucose are accounted for in the 2 3-carbon molecules of pyruvate. Now to get to this, there are 2 main phases in this reaction. I mean, it has to do with energy. May think of what they're called? 2 main phases having to do with energy. Alexis? Citric acid cycle is going to occur after this. All right, so we're not there quite yet. Sam?
Does it have to first go through phosphorylation?
Yeah, meaning.
Meaning an ATP attaches to glucose in organic phosphate group?
Okay, so yes, ATP is used.
NADH is like from NAD+, and it like can be either like NADH or FADH.
Okay, and so in this, in order to, let's go back to first what Sam said. You go through a series of reactions first, and I'm just going to go put some arrows here just to illustrate that it's not one big reaction, but you don't need to know the intermediate molecules that they go through the reactions to create. But at the end, we get from here to here. to get p-gal, all right, and there's 2 of those. And so I wanted to show you here that 2 p-gal, or another name for that in our notes and in our book, I may recall what it was called. They didn't call it p-gal, they called it something else. G3P, does it ring a bell? Not really. All right, so they're the same molecule, so I just wanted to point that out. So you have 2 of these p-gals here, all right, and this, if we look here, as Sam said, uses ATP. So I had a lot of people ask this question, so in the conversion of glucose to p-gal here, when you have an arrow going in like this and out on another arrow here, that means that during this reaction, so during the conversion of glucose to p-gal, that also 2 ATPs are converted to ADP. So what does that mean? ATP is a high energy molecule, has lots of energy in it, right? And so, so what happens is it phosphorylates the glucose, the molecule that was once glucose here, so you have a phosphate group here. So p-gal now has a lot of energy, more energy than glucose, because the energy that was once in ATP is now in p-gal, and so now we have ADP, which is lower energy than ATP. So therefore, that's what this diagram here was showing you. That's why this is up high here, is because they're relating potential energy, that p-gal has more energy than glucose. So glucose is a high energy molecule, but p-gal even has even more energy, and the reason why is because the use of the ATP, all right? And so in this here, how I want to show that here, when we use 2 ATP, so we're just going to say here, we're going to use 2 ATP, and it's going to be broken down into 2 ADP. So these guys have been phosphorylated. So we're using ATP. Because we use ATP, they call this the energy, what phase? Investment phase. So this is the energy investment phase. And because you have to invest some energy, right? Oh, yeah, that is supposed to say 2 AP, yeah, ADP. Huh? But she's not so nervous to point out when I do something wrong. I thought you're not so nervous to point out when I do something wrong. All right, so then the next phase, does anyone remember, now does this ring a bell, what happens in this conversion from here to here?
I just had a question.
Oh,
I'm still a little confused, like, what the arrow is showing with the ATP. Like, I'm still,
What, this arrow?
yeah.
Okay, so it means that in this reaction from here to here, or in the reaction to make p-gal here, that this reaction is going on, but to make this reaction, this is a endergonic reaction, it needs energy, and this is an exergonic reaction, so it's like 2 coupled reactions. So this exergonic reaction is driving this endergonic reaction. So therefore, the ATP is broken down into ADP in order to make this reaction happen. So this has been phosphorylated, that's where the P goes here, in converting glucose to p-gal. So it's like 2 reactions occurring simultaneously coupled together. Does that make sense?
Yeah, is the ADP though then just like released in the cell?
Yes. And so remember that ADP, this ADP that's released is gonna, remember the ATP cycle, if we add another phosphate onto it, it'll make ATP, which is what we're gonna do over here. Okay? All right? So we'll remake it into ATP eventually.
Wait, you said it's a, what reaction, sorry?
Okay, so this, from right here to right here is endergonic, needing energy, so ATP breaking down is the exergonic reaction. So this exergonic reaction is driving this endergonic reaction. So they're coupled together. So without that, we would never make any p-gal. All right, without the ATP. Okay. Okie doke. And so this next phase, so now we have a series of reactions to convert this P-gal into pyruvate, which is what we have here. And this, instead of using ATP, this makes ATP. And so how many total? Total is 4. All right, so that's what this is showing you here, is that from here to here, notice that this has high energy to lower energy. So, because the potential energy high to low, some of the energy that's stored in this molecule is used then to make NADH as well as ATP. Because to make ATP, that's an endergonic reaction. You have to have energy to put the phosphate back onto ADP to make ATP. So where does that energy come from? It comes from these reactions that are exergonic releasing energy. And so I'm going to just draw in general here, so it doesn't matter what phase here. What we need to know is that 4 ADPs become 4 ATPs. So we're making ATP in this, and so this is called, because we're making ATP, and we're making more than we put in, we call this the energy, starts with a P, payoff, right? So it pays off that we put a little bit of energy into it because we're getting more than we put in out of it, and so therefore we get a net gain, and the net gain is what from glycolysis? Net gain of ATP. Yeah, your net is 2, so we used 2 but got 4, so then our net gain is 2 ATP. So I'm going to put this down here with a little bubble around it here. This is big picture what we're getting out of this, and that so that our cells can use this ATP for energy. In our cell, is 2 ATPs a lot of ATP for cellular work, do you think? We talked about this in the notes, 2 ATP. No. All right. If this is all we did for every glucose, our cells would quickly die. All right, and so therefore, so really the purpose of glycolysis is to make a little ATP, but its real purpose is to slowly strip down glucose. All of these molecules that are intermediates are molecules that used to be glucose, and what we're doing is slowly taking the electrons off of that glucose molecule in the form of hydrogen. Every hydrogen atom has an electron. And so therefore, well, that's what we're doing here. So I haven't drawn that on here. What molecule is made that carries electrons in glycolysis that I haven't drawn here yet? Jesse? Yeah, the NADH. So the NADH right here is an electron carrier. And so, so in this process, this happens in the second step. We have 2 NADH pluses gets converted to 2 NADH. And so this, and all NADHs carry, I'm going to put here, carries 2 electrons. So we talked about that also in the notes. So the 2 electrons were once part of glucose. All right, and so it takes 2 hydrogens and makes the 2 NADH. So these are electron carriers. These NADHs, where do they carry electrons to? Where do these when I say it carries 2 electrons, so they're 2 electrons, what do they do with it?
They carry it in the electron transfer chain.
Yeah, they're going to carry it to the last step here that involves the electron transport chain. And so I put here in a little bubble here that 2 ATP is something that we get out that's good, that's a good end product. And then the NADHs, these guys, I'm going to put a little bubble around it, that's also a good, one of the main molecules that we want to get out of glycolysis. So the ATP is going to be used by the cell. NADHs are going to be used not here, all right, but these guys, I'm going to draw here an arrow to show them over here. They're going to use, and I'm going to draw in a little bit here, the third step, the electron transport chain. So the NADHs are going to be used in the mitochondria for the last step. And so And the last step is where the majority of ATP is made. So really the purpose of the previous step, like this step here, make little ATP and start the breakdown of glucose in stripping those electrons off. All right, so that is glycolysis. That's the end of glycolysis. So so before we get into the next step, let me give you the answers to the questions on this sheet that you did yesterday about glycolysis to make sure that we understand it now. So you had that model there, and number one, the molecule from food that's a reactant you should have is glucose, and B, glucose has 6 atoms, carbon atoms. 2, pyruvate has 3 carbon atoms, and B, they're made, there's 2 of them made, all right, for 2B. For number 3, does the process of glycolysis require an input of energy? Yes, that's the 2 ATP molecules in the energy investment phase that we just talked about. 4, propose an explanation for why the author of this activity put P-gal at the highest point in the model, and that is because that arrow here is kind of like a graph, like as the reactions occur, this gets converted to this, which gets converted to this. This is kind of like the y-axis here. They're trying to show you that it has higher potential energy. All right, so that means energy stored in the molecule. Number 5, does pyruvate have more or less potential energy than glucose? And so then we look here, all right, and so pyruvate here actually has, when we look at there, it's a lower point than glucose, all right, and so it has less potential energy than glucose. Number 6, the net production of ATP by glycolysis should be 2. 4 minus the 2 that were originally put in. 7, the electron acceptor is NAD plus is the electron acceptor. The publisher put NADH, but NADH is what is made after the NAD plus accepts the electrons. All right, so it's actually NAD plus being the electron acceptor. And then lastly, number 8, the sources of the 4 inorganic phosphates that are added to the ADP molecules are the ones that produce. Let me show you this picture here. All right. Each one of these here, all right, the BPG here has 2 phosphates, and there are 2 of them, so those are your 4 phosphates. So notice that pyruvate doesn't have any phosphates. All right, and so there's 2 here, and so the 2 come out of those, and since there's 2 of them, that accounts for 4 phosphate groups. All right, and that is the end. All right, with that one. So then the next step is, this publisher calls it the link reaction. I call it the intermediate step. It doesn't really have an official name. Okay, so let's go over the what happens next. So this is the link reaction. So these pyruvate are going to, I'm going to pick a different color for the link reaction. So choose, I'm going to pick red. You can pick whatever color you'd like. All right, and those pyruvate that are the end of glycolysis are now going to come into the mitochondria. All right, so now the two pyruvate that were made are in glycolysis as the beginning of this link reaction. Now, what happens to the pyruvate at the end of the link reaction? What is made?
Acetylcholine.
What's the molecule that pyruvate is going to kind of be converted into? Grace?
Acetyl-CoA.
Yeah.
Acetyl-CoA and it's pyruvic acid.
Well, pyruvate and pyruvic acid are the same molecule. Let's just talk about that real quickly here. Pyruvate, this part right here of it has the carboxyl group, the C with the double bonded O, and the O here. And the carboxyl group usually has the H here, but that's what the carboxyl group makes the molecule it's attached to acidic. And so what's happening here is if right now, this, as it's written right now with the H on it, it's pyruvic acid. But if it's already acted as an acid and gave up the H pluses, so then this is now O negative, it's called pyruvate. So pyruvate and pyruvic acid are the same molecule, it's just whether or not it gave up the H plus or not. Does that make sense? Okay? All right, and so, so we have the 2-pyruvate, and this gets converted and into 2 acetyl-CoAs. The CoA stands for coenzyme A. The 2 acetyl-CoAs are what the end product that's going to enter into the next major step. But there is some things that happen here in this reaction of pyruvate to acetyl-CoA. And so, and let's label this. They don't really call this a step, so it's not a real step, but let's call this the link reaction or the intermediate step. So oftentimes when they label steps for cellular respiration, they just skip over this one as a step. I'm not certain why, but they do. They don't have an official name or call it step 2 or anything like that. All right, with this, you do get something that's useful for this process of cellular respiration. What is it? So here's pyruvate to acetyl-CoA. What's made that's going to be useful in this process of cellular respiration besides the acetyl-CoA? What's going to be useful?
Was it NADH?
Yeah, it's the NADH. So we're in this process of converting pyruvate to acetyl-CoA, a couple of hydrogens along with the electrons are stripped from the intermediate there, and you get NADH. And so from this, I'm going to go ahead and draw, for simplistic sake, because for some room and so on, I'm going to assume now that every time an NADH is made, that you know it's made from an NAD+. So I'm not going to draw the reaction, I'm just going to show what's coming out of it. Does that make sense? Or should I keep it the same? Is it going to confuse everybody? Keep it the same. Okay. All right. So in this reaction here, NAD+ comes in, and since there's 2 pyruvates coming in and 2 acetyl-CoAs, then this happens twice then too. So I'm going to put a 2 in front of NADH or NAD+ and gets converted to 2 NADH. So that says 2 NAD+ gets converted to 2 NADH, kind of just like we did here with glycolysis. And this is, I'm going to put a little bubble around it again. An important product out of the link reaction. And this, because I'm going to draw some more here, I'm going to go ahead and put it up like this. And we're going to draw some things with the electron transport chain here later. So this carries electrons. It's an electron carrier that's going to carry it to that 3rd step with the electron transport chain. So we do get something useful out of that besides the 2 acetyl-CoAs. And then we also get some waste product. And so the waste product is CO2. So I'm going to go ahead and draw from this as well, the waste product coming off of here. And there's 2 CO2s. And I'll put in parentheses that this is waste. This is why we breathe out CO2. So this is 2 of the CO2s that are coming out here. That's why CO2 is on the right side of that equation. Okay, and that's the link reaction. And so we just get a little bit more electron carriers there. So now the acetyl-CoA goes where next? What does it enter? Acetyl-CoA enters the next step called the Krebs cycle or citric acid cycle. Before we go over that, I want to just go over the answers to the link reaction here on this. All right, so number 9 you should have is the the matrix of the mitochondria. So matrix, that's the inner membrane inside the membrane of the inner membrane here. It's all the space inside here, so that's where that reaction is happening. You need to know matrix, not just inside the mitochondria. Because if you say inside the mitochondria, it could mean the inner membrane space, all right, or it could mean the matrix. So number 10A. And asked you, I had a lot of questions on 10 yesterday. Is the pyruvate molecule likely to move across the mitochondrial membrane by diffusion? And it says include a comment about polarity. So when looking at pyruvate, pyruvate that has a charge on it. It has a negative charge there. That means it's hydrophilic, meaning that it's going to interact with water. And if you recall membranes, so when pyruvate enters here, it has to get across the outer membrane and the inner membrane. The outer and inner membrane have a construction very similar to the cell membrane, meaning that they are a lipid bilayer with proteins embedded. So simple diffusion, just regular simple diffusion, is through the lipid bilayer. So pyruvate, do we think that pyruvate can get through the lipid bilayer? It cannot. What kinds of things? So this is taking information from previous chapters, and what kinds of things can get through a lipid bilayer? Well, the major interior part of the lipid bilayer is hydrophobic. So hydrophobic things can get through, or very, very small polar molecules, and pyruvate's not that small. So therefore, but pyruvate needs to get into the mitochondria. So how do you suppose that it gets through? How do molecules get into the cell that can't get directly through the lipid bilayer? Michael? Yeah, protein carrier. So I'm going to go ahead and draw that in here. Here's a protein. All right, so it's getting through. There are proteins that they can diffuse through, and it is but this is facilitated diffusion. All right, so it is being helped along by these proteins. So then number 11, when the pyruvic acid is decarboxylated, what molecule is removed? That's CO2. Number 12, coenzyme A carries the remainder of the pyruvate to the site of the Krebs cycle. A, what is the name of the decarboxylated pyruvic acid? That is acetyl. Or B, there are 2 carbon atoms. C, the molecule that's produced is acetyl-CoA. And D was the one that it didn't show there, so I had you skip that. It was a typo on the picture there. 13, has any ATP been used or produced during the link reaction? Nope. 14, the high energy molecule that's been produced is NADH. So NADH is produced. And lastly, 15, how many of each? So how many acetyl-CoAs are there made? You should have 2, carbon dioxide 2, and NADH 2. All right? And so that's the answers there. So now we're on the Krebs cycle. 15? So for acetyl-CoA, there were 2 made. And I asked CO2 when there's 2, and NADH is 2. All right, so now we're on the Krebs cycle. Let's pick a different color. Let's see here. I'll pick blue, I suppose. Alrighty. So, so here we have acetyl-CoA. Now I'm going to draw it like this. This is acetyl-CoA. I'm going to start in a different color because now this was the product of the link reaction, and now it's going to be the first reactant of the next one. So I'm going to draw it like here, coming in to acetyl-CoA. And this enters into the Krebs cycle. And the Krebs cycle is a cycle meaning that, and I'm just going to draw a circle like this, because we don't need to know all the intermediates, all right? Like, so this shows you some of them, like citrate, and alpha-ketoglutarate, and succinate, and oxaloacetate. You don't need to know those names. And so what you need to know is that this is a cycle. So I'm going to call it, let's put citric acid or Krebs cycle here. I'm running out of room there. You can write cycle there. So what does a cycle mean? What it means is that the molecule you start out with, you reconvert and end up with. So therefore, oxaloacetate is the first molecule that comes and greets and reacts with acetyl-CoA. So right here, all right. And then it goes through a series of reactions and eventually ending up with oxaloacetate again, which can react with another acetyl-CoA, and the cycle can occur over and over and over again. So, so remember that the purpose of cellular respiration is to make ATP. Most of the ATP is made here in the third step. It's not made in these previous steps here. So Krebs cycle doesn't make a lot of ATP. Does it make some? Yes, absolutely. So here we see in this process here, some ATP is made. So in here somewhere, it doesn't matter where, we get 2 ADP gets converted to 2 ATP. All right, and this is, again, important things here. I'm gonna, the 2 ATP here, I'm gonna put down here. So these are the 2 ATP. All right, so we get, so far we've got 4 ATP made. All right, so again, 2 ATP, not a lot, so that's not really the purpose of the Krebs cycle. What do you think the purpose is of the Krebs cycle? It's not to make ATP. Liam? That's right, it's to make those electron carriers. NADH, and I introduced a new one, FADH2. So it's to further break down glucose, stripping the electrons from glucose and making these electron carriers so that these electron carriers can take the electrons to the electron transport chain where the majority of your ATP is going to be made. So that's what we have here. So you have NADH made as well as FADH2. All right, so in this here, we have and I'm going to, I just wanted to come out of this to show you that I'm not going to do it in all different steps. Like here they show it here, here, and here. We just need to know the big picture, that how many NADHs are made total. NADHs. How many times does the Krebs cycle happen? Once for every acetyl-CoA, right? So it happens twice. So here we have 1, 2, 3 NADHs. So it happens twice. So we have 6 NADHs coming off of here. So 6, I'm going to go here to this here. So we're going to have 6 NADHs coming off of there. And then I introduced a new electron carrier, and that was FADH2. And FADH2 also carries electrons. How many of those are made? 2. All right, so I'm going to put 6 NADHs on that same arrow. I'm going to draw 2 FADH2. So that's the main purpose there of that cycle, is to get those electrons out of those molecules that used to be glucose and get them to the electron transport chain. In this process of this series of reactions here to cause those things to be made, we get some more waste products. We get CO2. So the NAD plus, I can draw it in here. So we're also going, do you want me to draw it in here, the 6 NAD pluses, or are you just asking in general where they come? From, they're always recycled. So what happens when we get over here to the NADHs drop off their electrons, they're going to become NAD plus again, and then the NAD plus can come over here and become NADH, drop off its electrons, become NAD plus, and so they're cycled around and around. So they're literally just like a little shuttle, carrying the electrons, dropping them off, going to get more, dropping them off, and so on and so forth. So they're used over and over again. Kind of like ADP and phosphates to make ATP. You make ATP, put the phosphate on the ADP, you break it down, you put it back on, you break it, you know what I mean? All right, so, so that's where it comes from. All right, and so, any other questions on that? So then I was talking about the waste here. CO2 is made, and so you can see that these molecules, like from citrate to alpha-ketoglutarate, is one less carbon. That's because the carbon is released as CO2. And from here to here, it's one less carbon. That's because it's released as CO2. And so therefore, how many CO2s do we make or lose in this case? 4. There's 2 here, but then it happens twice, so you get a 4. So I'm going to go ahead and draw over here by these 2 CO2s. We get 4 CO2s that are a waste product. And lo and behold, here's our 6 CO2 coming out. All right, so those carbons used to be part of glucose, but now they're a part of CO2. All right. So now we're on this last part here where all these electron carriers go to drop off their electrons in this electron transport chain. So we're going to do a different color. All right. So electron transport chain I'm going to do in green. Where's the electron transport chain? Where am I going to draw this? In my picture here.
The membrane of the state.
Okay, yeah, these folds in the inner membrane. All right, so this is the electron transport chain. And remember, the electron transport chain have different molecules, and they're kind of clustered together into groups, and there's 3 of them. So I'm going to draw 3. And that they have these other ones that are kind of in between them. I can show you. I want to relate this to our picture in our notes so that you can see the connection. Hold on, I thought I had it open. I must have closed it out of the last hour. Let me show you the picture here. So my computer's kind of slow. All right, this last picture right here. All right, do you recall this picture in the notes? Here, so this picture is this picture here. So I forgot to do the one other little guy here. And so this is the electron transport chain. And so this is where it's at here, okay? And so the electrons, all these electron carriers are going to drop off these electrons. So, so one of the electron carriers is NADH. So we have them coming from all these different spots here. So I'm running out of colors here. I'm just going to do an NADH, let's say, from the citric acid cycle. So here's my NADH carrying how many electrons? 2, right? It carries 2 electrons. So what happens is the NADH drops off of its 2 electrons. So we're going to put 2E negative, meaning 2 electrons there. And those electrons, when the NADH drops off the 2 electrons, it's not going to be NADH anymore. It's going to be, when it does that, it's going to be now NAD plus and an H plus. It's gonna, because the H here is, one of the electrons here is gonna go to the electron transport chain. Remember, a hydrogen atom is a proton with an electron spinning around it. So therefore, when it gives the electron to the electron transport chain, it becomes H plus, all right? And so that's why it goes back to NAD plus, all right? All right, and going back to Alyssa's question, this NAD plus has done its job. It's carried the 2 electrons to the electron transport chain. This NAD plus can come over here and pick up more electrons, or come over here, all right, and so on. All right, so it goes, the NAD plus is always just going back, going to get more electrons, coming back, dropping them off, and so on and so forth. These electrons are now going to go through the electron transport chain. So they're going to go from molecule to molecule. So in blue here, I'm just going to go here are my 2 electrons moving through the electron transport chain. These are different molecules that it's moving to and from in the electron transport chain. Can somebody tell me why is it that the electrons move from molecule to molecule? Peter?
Is there a hydrogen ion gradient?
There is, that comes in just a minute here, all right? So before we get to that, you're right, it has to do with these electrons moving the hydrogen ion gradient, but let's talk about why they're moving through the electron transport chain in the first place. Each molecule is, down the chain, more what than the previous? Mark?
It gets more electronegative.
It gets more electronegative. So therefore, let's remind ourselves what electronegativity means. It means that it attracts or draws electrons to itself. So this first molecule has electronegativity and is higher than NAD+, so the electrons go to the first molecule in the electron transport chain. When that molecule gets the electrons, so I'm the first molecule in the electron transport chain, when I get the electrons, what am I? Oxidized or reduced? I am reduced. Now, the next molecule is even more electronegative, so the electrons don't want to stay with me. So then they go to the next molecule. That next molecule has been reduced. I have been oxidized. Then the next molecule draws those electrons to it, and it becomes oxidized and reduced. So this is a series of redox reactions going through here, where molecules are being oxidized and reduced, and they're moving from one to the other because the next one down the chain is more electronegative. Where you get to the end of the chain, and the final electron acceptor, or the most electronegative molecule, is what? Final electron acceptor. It's not actually part of the electron transport chain, but it is way more electronegative than even the last protein in this. Sam? Oxygen, O2. All right. And so oxygen is that final electron acceptor. So I'm going to put here final E acceptor. So that means it's the most electronegative. And what happens is when the electrons come over here with oxygen, they don't just meet up with oxygen. They meet up with some of the H pluses that are found in the matrix here. So therefore, we have H pluses plus the oxygen plus the electrons actually make what molecule? Water. And so this is our water at the end. Let's talk about why that is. H pluses that are found here from these reactions here, H pluses are in the matrix. You have H pluses and these electrons coming through. Do you see that if you have an H plus, is really a hydrogen missing an electron? So then the electrons come through, an electron meets with the H plus, and you get an H, all right? And then the other electron, I should put your 2 H pluses, meets another H plus, you get an H. So there's your so you get hydrogen atoms being made. And those hydrogen atoms are going to meet up with the O2 to make H2O, all right? And so therefore, this is the H2O here, okay? So that's the H2O that's coming off of here. That's the electron transport chain. What did I draw that's real important about the electron transport chain? When those electrons are moving through, what happens? That I totally missed purposely, but that I didn't draw. What's going on? Grace?
Pumping the hydrogen.
All right, yes, pumping the hydrogen from where to where? Where are these hydrogen going? Peter?
Isn't it the inner membrane space?
The inner membrane space. All right, so so these H pluses, some of them, as these two electrons go through this protein complex, then it's going to cause. Let's see, do I want to do? I'll just trying to decide if I want to do a different color because so it all doesn't blend in together here, but that might be a little confusing here. So the H pluses are going to be pumped as the two electrons move through here. H pluses are pumped. As the electrons move through this complex, H pluses are pumped. As they move through this complex, H pluses are pumped. All right. When I say pump, what does that mean? Energy is being used, right? Now, there's actually no ATP being used here, and so the energy comes from the redox reactions. I said that as it's becoming reduced and then oxidized, energy is released, and that's the energy to pump the H pluses. From low, the pumping is assuming that it's going from low to high concentration. All right, so I'm going to leave, we're getting to the end of the hour here. I'm going to leave you with this. I want you to think about why is it that we want a high H plus ion concentration in the inner membrane space? All right, so why is that important? Yes, yeah. I will finish going over the answers to the packet tomorrow. I got a little carried away with the electrons, unfortunately. You know what, I meant to, yes, it should be 4 H pluses with the O2 to make 2 H2, yes. Our book says 2 H pluses but plus a half of an O2, which would really be an O. So I actually my O. All right, so I'll fix that. Remind me tomorrow and I'll say that to the class.